Welcome back to another Research Connection. Today I am here with Dr. Ravi Kulkarni. He is an assistant professor in our Population Health and Pathobiology department focusing on poultry immunology and microbiology, and we are going to talk to him about a recent grant he was awarded with the USDA looking at poultry health, specifically turkeys, and a probiotic-based vaccine. So your research is specifically in immunology and infectious disease, and you focus a lot on antibiotics and finding alternatives for poultry flocks. Can you talk about the importance of that in our society and why it's such a focus of research right now?
Yeah, Sarah, the most important point is like amongst the food animal production, poultry is playing a very important and integral role in terms of providing, best source of protein. And we also know that, the global population is going up, and then there will be more and more demand for food animal production. So currently, it's also like, as there is intensified production of poultry, there is also one factor that is coming up is in a big way is antimicrobial resistance. So in order to, reduce the spread of antimicrobial resistance, the industry has been moving towards no antibiotic-free farming or reduced antibiotics in the poultry production. So what happens is traditionally, poultry have been reared with the use of antibiotics at a subtherapeutic level in the feed so that most of the enteric infections have been kept under control, particularly the bacterial infections, so which could include Clostridium, Salmonella, Campylobacter, and so on. So now when you are moving away from the antibiotic, then all these pathogens will have like a brilliant opportunity to grow in the intestine and produce the disease, right? Mm-hmm. So when we don't want to use the antibiotics, we have to come up with alternatives to antibiotics. In order to do that, my laboratory focuses on understanding the immunology and microbiology of these diseases, and then try to come up with alternatives such as using probiotics or making vaccines or integrating probiotics and vaccines in order to find a solution where the poultry can sustain without the use of antibiotics.
You were recently awarded a grant with the USDA that's focusing on a probiotic-based vaccine for turkey flocks. Can you talk a little bit about that type of immunity and how it works and how it's different than how we normally think of receiving a vaccine, which is, a shot in your arm? Can you just elaborate a little bit more on that in your project?
We are trying to make a vaccine that has two qualities into one. Probiotics are, we all know in the grocery stores, which we get it, right? Mm-hmm. So they are good bacteria. Mm-hmm. And then we also know that there are certain vaccine candidates. Then you can take the genes for the vaccine candidates from the vaccine mm-hmm. and then put them into the probiotic good bacteria. Then you can develop a two-in-one kind of vaccine, which is you will get the good bacteria probiotic effect, good effects, as well mm-hmm. as your vaccine will keep doing what it is supposed to. So ideally, the vaccines are targeted to prevent the disease, right? So we all are like very well educated in the vaccinology and stuff. And here, traditionally, if you think about a vaccine, we always think about shots, right? So it goes into the muscle or sometimes subcutaneously under the skin and so on. But when it comes to poultry, we cannot think about one, these are in thousands, and then you have big farm. And the only feasible or practically applicable mode of vaccine delivery would be something you can put it in the feed, you can put it in the water, so that it is a mass administration. All the birds can drink water, eat mm-hmm. feed, at the same time they get the vaccine too. So when this vaccine is all about Clostridial dermatitis. When we say dermatitis, we all know that the skin becomes reddened. But in this particular case, the causative agent, the bug that causes the disease is called Clostridium septicum. So these are anaerobic organisms in the sense that these bugs, these bacteria, they don't like oxygen. So wherever they grow, they produce some sort of a gas, and then you might have heard about gangrene, right? Yes. So it's basically in the skin, it creates an environment where there is a lot of dead tissue, and the bird becomes sick, and a lot of them die, and so on. So the whole idea is that if we can make a vaccine that can be administered orally, either in the feed or the water, and which can produce its probiotic health benefits, and at the same time they are protected, means they can prevent or they can resist this disease. So that's the whole concept that we pitched, me and my team, and then USDA liked it and we got the grant.
I understand that there are 3 stages to this project and a lot of different collaborations both on our campus at NC State College of Veterinary Medicine and on different campuses around NC State. Can you talk a little bit about the different stages of the project and your different collaborators on it?
Sarah, I always have believed that over the years, collaboration is the key to the research, success of a research program. And here at College of Veterinary Medicine too, there is quite a bit of support to build collaborative teams and then write grants and bring the funding and do the research. So in this case, as you mentioned, this project has like 3 stages, which comprises of certain expertise in immunology and microbiology, which my laboratory is involved, and then this work also involves a lot of molecular cloning work, where we have great expertise from CALS at NC State. Dr. Rudolf Barangu, who is a world-renowned expert in molecular biology and then CRISPR technology, and also Dr. Sarah O'Flaherty, she's a senior scientist there. And then this work also requires the aspects of poultry medicine and clinical parts of it, where Dr. Rocio Crespo here from the College of Veterinary Medicine, I call her a clinician scientist. And we also have Dr. Anil Thachil from the Rollins Animal Diagnostic Laboratory. So that's how we have built up this team to do this work in 3 stages. First stage is it involves identifying what the strains or the isolates of the good bacteria, which is the probiotic, which are specifically helpful in turkeys. The second stage is once we have these identified bacteria, then we would like to involve the molecular expertise from Dr. Barangu and Dr. Flaherty to put the genes, the vaccine candidates, into these probiotic and then make this vaccine as a 2-in-1, as I mentioned. And the third stage is where actually we have this laboratory constructed vaccine, taking it to the birds in case of turkeys to administer orally in the water or in the feed, and then see if this vaccine can work. At the same time, it can also help the overall health of the turkeys, wherein Dr. Crespo has expertise in the clinical monitoring of the birds and then pathology and so on. So this is how we have built up a multidisciplinary team for conducting the research involved in this project.